11.6.3 Chemiosmotic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemiosmotic hypothesis has been put forward to explain the mechanism. Like in respiration, in photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time, these are the membranes of thylakoid. There is one difference though. Here the proton accumulation is towards the inside of the membrane, that is in the lumen. In respiration, protons accumulate in the intermembrane space of the mitochondria when electrons move through the ETS, chapter 12. Let us understand what causes the proton gradient across the membrane. We need to consider again the process that take place during the activation of electrons and their transport to determine the steps that cause a proton gradient to develop. Figure 11.7. Number A. Since splitting of the water molecule takes place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoids. Number B. As electrons move through the photosystems, protons are transported across the membrane. This happens because the primary acceptor of electron, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side or the lumen side of the membrane. Number C. The NADP reductase enzyme is located on the stroma side of the membrane. Along with electrons that come from the acceptor of electrons of PS1, that is photosystem 1, protons are necessary for the reduction of NADP plus to NADPH plus H plus. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. बिकॉज ल्यूमन में H प्लस की कंसनट्रेशन क्योंकि बढ़ रही है, तो जितना ज़्यादा H प्लस का कंसनट्रेशन बढ़ेगा pH उतना कम होगा, pH डिक्रीज करेगा ल्यूमन के अंदर. Why are we so interested in the proton gradient? This gradient is important because it is the breakdown of this gradient that leads to synthesis of ATP. The gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channel of the CF0 of the ATP synthase. The ATP synthase enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called the CF1 and protrudes on the outer surface of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthase, which makes the enzyme synthesize several molecules of energy packed ATP. Chemiosmosis requires a membrane, a proton pump, a proton gradient, and ATP synthase. Energy is used to pump protons across a membrane to create a gradient or a high concentration of protons within the thylakoid lumen. ATP synthase has a channel that allows diffusion of proton back across the membrane. This releases enough energy to activate ATP synthase enzyme that catalyzes the formation of ATP. Along with the NADPH produced by the movement of electrons, the ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing CO2 and synthesis of sugars. 11.7. Where are the ATP and NADPH used? We learned that the products of light reaction are ATP, NADPH and O2. Of these, O2 diffuses out of the chloroplast while ATP and NADPH are used to drive the process leading to the synthesis of food, more accurately sugars. This is the biosynthetic phase of photosynthesis. This process does not depend directly on the presence of light but is dependent on the products of the light reaction, that is ATP and NADPH, besides CO2 and H2O. That's why this is also known as a light dependent reaction. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, the biosynthetic process continues for some time and then stops. If then light is made available, the synthesis starts again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourself. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that CO2 is combined with H2O to produce CH2O whole N or sugars. It was of interest to scientists to find out how this reaction proceeded, or rather what was the first product formed when CO2 is taken into a reaction or fixed. Just after World War II, among the several efforts to put radioisotopes to beneficial use, the work of Melvin Kelvin is exemplary. The use of radioactive C14, that is carbon 14, by him in algal photosynthesis studies led to the discovery that the first CO2 fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway. Hence, it was called Calvin cycle after him. The first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of CO2 fixation or whether any other product was formed in other plants. Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product of CO2 fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxaloacetic acid, or OAA. Since then, CO2 assimilation during photosynthesis was said to be of two main types: those plants in which the first product of CO2 fixation is a C3 acid, that is PGA, that is the C3 pathway, and those in which the first product was a C4 acid, OAA, that is the C4 pathway. These two groups of plants showed other associated characteristics that we will discuss later. 11.7.1 The primary acceptor of CO2. Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting, that is fixing CO2, would have 3 carbons of PGA? The studies very unexpectedly showed that the acceptor molecule was a 5-carbon ketose sugar, that is a ribulose bisphosphate, RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time and conducted many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2 carbon compound, ताकि वो एक CO2 का कार्बन ले करके 3 कार्बन कंपाउंड बना लेगा. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. 11.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understand very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathway, figure 11.8. For ease of understanding, the Calvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. Number 1. Carboxylation. Carboxylation is the fixation of CO2 into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where CO2 is used for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it as RUBP carboxylase oxygenase or a Rubisco. Number 2, reduction. These are a series of reactions that lead to the formation of glucose. The steps involve utilization of two molecules of ATP for phosphorylation and two of NADPH for a reduction per CO2 molecule fixed. The fixation of 6 molecules of CO2 and 6 turns of the cycle are required for the formation of 1 molecule of glucose from the pathway. Number 3, that is the last step, is regeneration. Regeneration of the CO2 acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps require 1 ATP for phosphorylation to form RUBP. Hence, for every CO2 molecule entering the Calvin cycle, 3 molecules of ATP and 2 NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecule will be required to make 1 molecule of glucose through the Calvin cycle. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. Which goes in, it includes 6 CO2, 18 ATP and 12 NADPH, and what comes out is 1 glucose, 18 ADP and 12 NADP. 11.8, the C4 pathway. Plants that are adapted to dry tropical regions have the C4 pathway mentioned earlier. Though these plants have the C4 oxaloacetic acid as the first CO2 fixation product, they use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way are they different from C3 plants? This is a question that you may reasonably ask. C4 plants are special. They have a special type of leaf anatomy. They tolerate higher temperatures. They show a response to high light intensities. They lack a process called photorespiration and have a greater productivity of biomass. इसलिए C4 plants are more beneficial than C3 plants. Let us understand these one by one. Study vertical section of leaves, one of a C3 plant and the other of a C4 plant. Do you notice the differences? Do both have the same types of mesophils? Do they have similar cells around the vascular bundle sheath? The particularly large cells around the vascular bundles of the C4 plants are called bundle sheath cells, and the leaves which have such anatomy are said to have cranch anatomy. Cranch means a wreath and is a reflection of the arrangement of cells. The bundle sheath cells may form several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick walls, impervious to gases exchange, and no intercellular spaces. You may like to cut a section of the leaves of C4 plants, for example maize or sorghum, to observe the cranch anatomy and the distribution of mesophyll cells. It would be interesting for you to collect leaves of diverse species of plants around you and cut vertical section of the leaves. Observe under the microscope, look for the bundle sheath around the vascular bundles. The presence of bundle sheath would help you identify the C4 plants. Now study the pathway shown in figure 11.9. This pathway that has been named the Hatch and Slack pathway is again a cyclic process. Let us study the pathway by listening the steps. The primary CO2 acceptor is a 3-carbon molecule phosphoenol pyruvate PEP and is present in the mesophyll cells. The enzyme responsible for this fixation is PEP carboxylase or PEPcase. It is important to register that the mesophyll cells lack Rubisco enzyme. This C4 acid OAA, that is oxaloacetic acid, is formed in the mesophyll cells. It then forms other 4-carbon compounds like malic acid or aspartic acid in the mesophyll cells itself, which are transported to the bundle sheath cells. In the bundle sheath cells, these C4 acids are broken down to release CO2 and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll cell where it is converted to PEP again, thus completing the cycle. The CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. The bundle sheath cells are rich in an enzyme ribulose bisphosphate carboxylase oxygenase Rubisco but lack pepcase. Thus, the basic pathway that results in the formation of the sugars, the Calvin pathway, is common to C3 and C4 plants. Did you note that the Calvin pathway occurs in all the mesophyll cells of the C3 plants? In the C4 plant, it does not take place in the mesophyll cells but does only in the bundle sheath cells. ये line important है. Next topic is photorespiration. 11.9 Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 plants, that is photorespiration. To understand photorespiration, we have to know a little bit more about the first step of the Calvin pathway, the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA, that is catalyzed by Rubisco. Rubisco, that is the most abundant enzyme in the world, do you wonder why? Is characterized by the fact that its active site can bind to both CO2 and O2, hence the name. Can you think how this could be possible? Rubisco has a more greater affinity for CO2 when the CO2 is to O2 ratio is nearly equal. Imagine what would happen if this were not so. This binding is competitive. It is the relative concentration of O2 and CO2 that determines which of the two will bind to the enzyme. In C3 plants, some oxygen does bind to Rubisco and hence CO2 fixation is decreased. Here the RUBP, instead of being converted to two molecules of PGA, binds with oxygen to form one molecule of phosphoglycerate and phosphoglycolate, which is 2 carbon, in a pathway called photorespiration. In the photorespiratory pathway, there is neither synthesis of sugars nor of ATP. Rather, it results in the release of CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. In C4 plants, photorespiration does not occur. This is because they have a mechanism that increases the concentration of CO2 at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down in the bundle sheath cells to release CO2. This results in increasing the intracellular concentration of CO2. तो ऑक्सीजन है ही नहीं कि वो Rubisco के साथ bind करेगा. In turn, this ensured that the Rubisco functions as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plants lack photorespiration, you probably can understand why productivity and yields are better in these plants. In addition, these plants show tolerance to high temperatures. Based on the above discussion, can you compare plants showing the C3 and the C4 pathway? Use the table format given in table 11.1 and fill in the information. 11.10 Factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plants, including crop plants. Photosynthesis is under the influence of several factors, both internal, that is plant, and external. The plant factors include the number, size, age and orientation of leaves, mesophyll cells and chloroplast, internal CO2 concentration and the amount of chlorophyll. The plant or internal factors are dependent on the genetic predisposition and the growth of the plant. The external factors would include the availability of sunlight, temperature, CO2 concentration and water. As a plant photosynthesizes, all these factors will simultaneously affect its state. Hence, though several factors interact and simultaneously affect photosynthesis or CO2 fixation, usually one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's 1905 law of limiting factors comes into effect. This states the following. If a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value. It is the factor which directly affects the process if its quantity is changed. For example, despite the presence of a green leaf and optimal light and CO2 conditions, the plant may not photosynthesize if the temperature is very low. This leaf, if given the optimal temperature, will start photosynthesizing. 11.10.1 Light. We need to distinguish between light quality, light intensity, and the duration of exposure to light while discussing light as a factor that affects photosynthesis. There is a linear relationship between incident light and CO2 fixation rates at low light intensities. At higher light intensities, gradually the rate does not so further increase as other factors become limiting. तो जो भी फैक्टर लिमिटिंग होगा ना उसी पर फोटोसिंथेसिस डिपेंड करेगा. तो लाइट जब तक लिमिटिंग है तो वो लाइट के डायरेक्टली प्रोपोर्शनल है, लेकिन जब एक मैक्सिमम लेवल पहुंच जाएगा उसके बाद जो फोटोसिंथेसिस है वो लाइट के डायरेक्टली प्रोपोर्शन नहीं रहेगा, क्योंकि कोई और ऐसा फैक्टर होगा जो लिमिटिंग बन जाएगा. तो फिर फोटोसिंथेसिस जो है वो वाले लिमिटिंग फैक्टर पर डिपेंड करने लगेगा. यही इसमें एक्सप्लेन किया है. दैट इज व्हाट ब्लैकमैन्स प्रिंसिपल. What is interesting to note is that light saturation occurs at 10% of the full sunlight. Hence, except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point causes the breakdown of chlorophyll and a decrease in photosynthesis. 11.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere, that is between 0.03 and 0.04%. Increase in concentration up to 0.05% can cause an increase in CO2 fixation rates. Beyond this, the levels can become damaging over longer periods. The C3 and C4 plants respond differently to CO2 concentrations. At low light conditions, neither group responds to high CO2 conditions. At high light intensities, both C3 and C4 plants show increase in the rates of photosynthesis. What is important to note is that the C4 plants show saturation at about 360 microliter per liter, while C3 responds to increased CO2 concentration and saturation is seen only beyond 450 microliter per liter. Thus, current availability of CO2 level is limiting to the C3 plants. The fact that C3 plants respond to higher CO2 concentration by showing increased rates of photosynthesis leading to higher productivity has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads to higher yield. 11.10.3 Temperature. The dark reactions, being enzymatic, are temperature controlled. Though the light reactions are also temperature sensitive, they are affected to much lesser extent. The C4 plants respond to higher temperature and show higher rate of photosynthesis, while C3 plants have a much lower temperature optimum. The temperature optimum for photosynthesis of different plants also depends on the habitat that they are adapted to. Tropical plants have a higher temperature optimum than the plants adapted to temperature climates. 11.10.4 Water. Even though water is one of the reactants in the light reaction, the effect of water as a factor is much more through its effect on the plant rather than directly on photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaves wilt, thus reducing the surface area of the leaves and their metabolic activity as well.